Show me by hand. Dear learners, you're most welcome once again to our lesson in biology, which is a continuation of the topic of homeostasis, which we have learned previously in our lessons. Now, just remind ourselves, remember we said in homeostasis we are looking at the maintaining of the internal environment at a constant state, regardless of the environmental conditions. Now, we studied about fluid formation, tissue fluid formation. We also went ahead to look at the most important organ in our body, which is the liver, regarding its functions. We looked at one of the most important functions of the liver, and that is carbohydrate metabolism. Dear learner, we looked at this in our previous lesson, and just to remind ourselves, we looked at in sugar regulation, we must first observe what is the norm, what is the set point. Remember when you looked at the components of homeostasis, set point is one of the components that must be considered in discussing homeostasis. Now, when it comes to carbohydrate metabolism, previously we looked at sugar regulation. In sugar regulation, we must first look at what is the norm, what is the set point. And we discussed and say that the set point for sugar in the blood is 90 milligrams per 100 centimeter cubed of blood. Then we went ahead to look at what if the sugar level goes above, or that is above 90 milligrams per 100 centimeter cubed. In that case, we said the condition of the body becomes hyperglycemia. Hyper means it is up above the set point, which is 90 milligrams per 100 centimeter cubed. Then, dear learner, we say that in this case, the detector for the excess blood sugar is in the pancreas. And these cells, we call them the islets of Langerhans, which are stimulated to secrete a hormone called insulin. So in this case, insulin is secreted into the blood and then flows to reach its target cells, which are the hepatocytes. The hepatocytes are the liver cells. So in this case, the liver cells, the hepatocytes, under the influence of insulin, will be able to carry out a metabolic activity which will convert the excess blood sugar into glycogen, and it is stored. Then we looked at glycogenolysis. Lysis means to break down. In this case, still the same islets of Langerhans, the cells of the pancreas are the detector. And they respond by releasing a hormone called glucagon. Once again, the glucagon is transported to the blood, in the blood, all over the body to be there, but it must be reaching the target cells. That's when its influence will be there. In this case, the target cells still are the muscle cells, but largely the liver cells. So the other stored glycogen, as we looked before, will be stimulated to be broken down and to increase the blood sugar, which is now free. A continuation of that is what we're going to stem our topic today, part of our topic today, and that is we are going to look at another process of carbohydrate metabolism, and that is gluconeogenesis. So we have gluconeogenesis. If you can break down this term, dear learner, we have gluco. That means the formation of glucose, in this case, from non-carbohydrate products. What are these non-carbohydrate products? For example, we have proteins. Okay? And when does this gluconeogenesis occur? It occurs in the absence of glucose. There will be no free glucose in blood. There will also be no the glycogen, the stored sugar in the cells. So it occurs, for example, when the body is starving, when the glucose is not, the source is not available, you have not eaten, or even when someone is fasting for long and does not take any meal. So, for example, these products, non-carbohydrate products, where the glucose will be converted, is one of them is protein and lipids. These are the major ones. Briefly, how does this give us the how does this give us the glucose? Now, for example, if you look at the protein, we know very well the building block of a protein is amino acid. So in this case, from the amino acid, in this case, what exists into the blood is the amino acid, not the protein as per se. The protein itself is a bigger molecule. It's a polypeptide. Now the amino acid, if we can use just a word equation, then we shall look at the chemical one. So we shall have the amino acid undergoing what is called deamination. The deamination, that means it involves it reacting with oxygen. Okay, it will give us a carboxylic acid, silic acid, and of course plus ammonia. We shall look at the fate of that ammonia later. But our major concern is this carboxylic acid. So this carboxylic acid will undergo respiration in the mitochondria to yield energy. In this case, this carboxylic acid is a product of which is like a carbohydrate. If we look at it in terms of the equation, that means we shall have the amino acid. We have the amide group. Okay, amine group, which is that one. In presence of oxygen, so this is deamination. It will give us the carboxylic acid, have the oxygen, the R, and yes, COOH, and of course now plus the ammonia. In deamination, what occurs is this amine group is removed. And therefore, our major concern for this part of gluconeogenesis is this. So this product goes into the Krebs cycle, that is in respiration, in this side the mitochondria, and energy is formed. Then when it comes to lipids, I'm back to the lipids undergoes beta oxidation, to give us the fatty acids, and also you're going to get glycerol. This glycerol, if you can remember very well in the topic of respiration, the glycerol will combine with the phosphate, form glycerol 3-phosphate. Okay, and the glycerol 3-phosphate undergoes a stepwise breakdown, what we call glycolysis, and to yield more energy. Then when you come to these fatty acids, the fatty acids again will undergo stepwise breakdown in the process of Krebs cycle, that is inside the mitochondria, to yield more energy. In this case, the genesis, we have built up a carbohydrate that will be broken down to release energy in absence of a glucose molecule. That is what we looked at. So dear learners, questions can come in this. And I would like you to note some of this down from what you've just looked at about carbohydrate metabolism. One of the questions you can get is to describe the process of blood sugar regulation by the liver. Describe the process of blood sugar regulation by the liver. Dear learners, let us continue with functions of the liver, just in connection with this process of deamination. And apart from this carbohydrate regulation, we also have some other process of metabolism. And in this case, it is protein. So you can term it as protein metabolism. So number 2, we have protein metabolism. How does this one occur? Somehow, dear learner, you must have realized we have mentioned a protein here. So how does this one occur? How are proteins regulated in our body? Metabolism. Now, when it comes to what we refer to as the set point, in this case, the set point for a protein varies from one body to another. Why? Because different bodies have different body size and different body needs. So it's not like blood sugar and body temperature. So it varies. However, there must be its regulation. Proteins is one of those nutrients which are not stored in the body. We only take what is necessary, what is required. The excess one must be eliminated from the body. So we have regulation of amino acid. In this case, it is the amino acid which is regulated. Amino acid that is regulated. Just as the same way we looked at carbohydrate and we mentioned the glucose. So in this case, the excess amino acid are not stored as we said before, but rather the supply is get rid. Where? In the liver. So we have it involves the process of deamination, as we are going to look at, where in deamination, previously we looked at deamination, so it involves, there must be deamination. Dear learner, just remind ourselves once again, deamination. We are removing the amino group from the amino acid. And this amino group, the amino group will form what is called ammonia, and the ammonia reacts with ornithine, which is a protein present in the liver cells, that is the hepatocytes, which undergoes a series of reactions in the ornithine cycle, at last to form urea, which is again transported by blood and eliminated in the liver. Now let us look at in details what really occurs in this amino acid regulation. So we said the first process is deamination, and in deamination, as we looked at, as I am going to repeat, we have the amino acid, okay, so reacting with oxygen, so that will form the carboxylic acid. We have a carboxylic acid. Let us first write it in words for our better understanding. Carboxylic acid, and then plus ammonia. Let's look at in terms of the chemical equation. However, don't get scared of the chemical equations. This is for our better understanding. Remember, biology and chemistry, they are related somehow. Now, let's look at this amino acid. The formula is, you can also try to write it down for yourself. And then you have this, R here, COOH, and then we have the H. This is a simple formula for the amino acid. So deamination reacts with oxygen gas. That means deamination requires oxygen for us to form this carboxylic acid and plus the ammonia. Once again, in deamination involves the removal of the amine group. And amine group combines with the hydrogen. From this, remove this ammonia. Therefore, in this case, it is this. So if you are, that is the formula. In this case, it is this ammonia that will undergo now in a series of reactions in a cycle called the ornithine cycle. So let's look at the ornithine cycle. Or me. ornithine cycle. Briefly, we can look at the ornithine cycle, where you're saying the ammonia, in this case, will react with the ornithine, which is always present in the hepatocytes, in the liver cells. But in this reaction, it also involves the combining between carbon dioxide and also water is involved to form another compound, which is also a protein called citrulline. In this case, in that process, here water is taken in, but at the same time also water is removed. So therefore, water is not stable in that process. Now, the citrulline is again undergoes ammonia, picks the ammonia, other molecules of ammonia. And this process where ammonia is picked, also we are going to say that the water molecules be given out to form another protein in the hepatocytes called the arginine. So here you have the arginine. Then later on, this arginine will again be hydrated. That means combined with the water. Okay, to form, to give us now the urea, and then other product will be recycled back so that we continue to have. So since the ornithine will be resynthesized, that means that's why this process is called the ornithine cycle. Now this urea here is the one that will be transported in blood until it reaches the kidney. Now in the kidney, that is when now it will be eliminated from the body. That is what occurs. So in this case, we have said that the excess amino acids, which are the building blocks of protein, they are not stored, but rather they are broken down in a process called the ornithine cycle to urea. In this case, the urea will be carried by blood into the kidney to be inert as urine. We are going to look at that process also. Now, that is how proteins are regulated. We also have another form in which the proteins are regulated, and that is through transamination. So we have what's called transamination. Dear learner, in transamination, it involves the transfer of the amino group. What is the amino group here? The amino group is this NH2 from the amino acid. We have the R here, then H, COOH. So the amino group is this one. So when this amino group is transferred to another organic compound, it will form an amino acid. So it transferred. So that means you can put a plus, the organic compound. It ends up forming an amino acid. In this case, this amino acid will end up is referred to as non-essential amino acid. Remember, we have two types of amino acids, the essential and the non-essential. This non-essential amino acid, which is non-essential, the non-essential is formed in the body. That means in the body cells. And it is required in small amounts. And what is its function? This is the one used in information. It is for export in the cell. If you can remember, we talked about the proteins which involve information of enzymes and hormones are these ones. the non-essential. Then the essential ones, how do we obtain them? Dear learner, yes, I know you have written it down. So the essential amino acid, we obtain them from the foods that we eat, especially the products of plants, meat, fish, milk, and so on. That is how we have the transamination occurring. Again, dear learner, remember the major purpose of our studies here is to pass the exam. So again, I keep on reminding ourselves, we need to be noting the most important parts that will help us to understand our notes. But again, in this, you can get questions. They are in line with some of the questions that can come in this case. And some of these questions are to differentiate deamination and transamination, or to distinguish deamination and transamination. Then you can also get this question, which I request that you write it down. That is to describe the process of urea formation in the liver. Describe the process of urea formation in the liver. Thank you. Let us continue, and we also look at other functions of the liver. To remind ourselves, dear learner, I said that the liver performs more than 1,000 functions. Only that for purpose of our studies, we study a few of them. And we just emphasize again a few for purpose of our studies. So we also have other functions of the liver. Other functions of the liver. And what are these other functions of the liver? Remember, we say that the liver performs a very important function of getting rid of the old red blood cell. That means the breaking down of worn out red blood cells. How can this one be put more clearly? Red blood cells have a lifespan of 120 days. Of these 120 days, that is approximate 3 months. So note that all of them are formed at once. That means that even as the ones are formed, others are also being broken down. So when it comes to the fetus, the red blood cells are broken down in the liver, in the fetus. Now, how is this one possible? It is possible because we have the Kupffer cells. It is possible because we have the Kupffer cells. These Kupffer cells, they are phagocytic. They also have macrophage. That means they are capable of breaking down their lysis. They break down the red blood cells. And the product that we get here is what we call bilirubin. This bilirubin is a major product in formation of bile, which is also one of the functions of the liver. So that means also another function is production of bile. Bile. That means the bile juice. So I'll just to emphasize that one. The bile pigment, the bilirubin, okay, come from the red blood cells when they are broken down. That means production starts with the breaking down of the red blood cells. And this bilirubin is the reason why the food that we eat is not the same, the color of the food that we eat. When this bile is segregated on the food after digestion, that is after the breaking down from the stomach, that is the duodenum, the color changes due to the bilirubin, the pigments. Also, we have other functions which involve detoxification. Talk about number 3, detoxification. Dear learner, to detoxify means to break down a poisonous material into the one which is harmless. And how is this one possible? The liver has numerous organelles, and one of the one, and it's very important, one is mitochondria, and most important we have the smooth, presence of the smooth endoplasmic reticulum. Why this smooth endoplasmic reticulum? Just remind ourselves, the smooth endoplasmic reticulum are organelles involved in fat metabolism. In breaking down any poisonous material, any toxic material in your body, one of the mechanisms is or involves the lipids. Therefore, the synthesis of lipid is very important, and lipids are synthesized in the smooth endoplasmic reticulum. That means the products are formed there, and then in the Golgi apparatus, then excretion are produced. Then we have other function, that is the synthesis of the plasma protein. Synthesis of plasma protein. Another function number 4, we have synthesis of the plasma proteins. What is the importance of this plasma protein? Its importance is maintaining osmotic pressure of blood. We know very well that one of the major factors that allows the maintenance of the osmotic pressure of blood is the mineral salts, the sodium, the chlorides, the potassiums, and so on. But also these plasma proteins are important. And where do they come from? From amino acids. How does this one occur? Now it occurs when we have processes such as in regulation of proteins, and the amino acids which are formed in this one, in this case, they include the albumin. So these ones include albumin. Albumin, which is involved in transportation, or it's a transport molecule. We also have globulin, which is a molecule also that is concerned in formation of hormones. We have prothrombin. Dear learners, you can remember very well that prothrombin is involved in blood clotting. So prothrombin and fibrinogen. These are the important proteins which are synthesized from amino acid and their plasma proteins. Other functions, as we have said earlier, that means is the production of bile, okay? And another one is the storage of fat soluble vitamins. Storage, so we have storage of fat soluble vitamins. What are these fats? We have vitamin A, vitamin D, vitamin E, and K. You must have realized, even in a home setting, you are advised that try to eat vitamins. Try to eat liver, okay, as being a source of vitamins. The reason being, in each cells they are stored vitamin A, D, E, and K. Now, not only that one, the liver also is a source of mineral ions. That means it also stores mineral ions, such as iron. Again, here to remind us of the advice which most doctors give, that when your blood level is low, try to eat liver. Why? Because it contains iron, which is a key component in formation of the hemoglobin. So it stores iron, potassium, copper, zinc, among others. It also stores blood. So please, dear learner, to keep on noting down. Also stores blood. So in relation to its structure, what are these uh, uh, adaptation that it has that allow it to store blood? So 7 have storage of blood. So the key factor that allows the liver to store blood, one of them is it is elastic. But most important, because the blood vessels in it are elastic, they can expand to accommodate more blood, especially in situations where the bodies are stressed. However, these blood vessels can also vasoconstrict under the influence of hormones such as adrenaline in case of stress or when the body is more active, so that more blood can flow in other parts of the body. So therefore, in this case, we are looking at the elasticity of the blood vessels, which can expand when the body is at rest and store more blood. So we say the functions are quite many, but we're just mentioning a few which are very important in our studies. It also destroys uh, destroys uh, all hormones, especially the sex hormones. Destroys the sex hormones after exerting their effects. After their effects. For example, testosterone. For example, hormone testosterone. This hormone must be destroyed. If it doesn't, it is not destroyed, it will continue exerting its effect. That means ejaculation will be prolonged. So it must be destroyed. Then another one we have is in the regulation of lipids. Regulation of lipids. You can call it lipid metabolism. Dear learner, remember when we talk about the metabolism, it refers all the biological processes involving breaking down and building up. So in this case, the excess cholesterol, which is a lipid in blood, is excreted into the bile. And what does it form? It must be excreted so that it avoids its accumulation. Its accumulation in the blood will lead to the it sticking onto the blood vessels, and in this case can result into condition of what you call, for example, coronary thrombosis. So when it sticks into the walls of the blood vessels of the heart, this cholesterol will keep on increasing, and by then, as it is sticking onto the walls of the blood vessels of the heart, therefore it will lead to the reduction in the surface area of blood that is passing. And this results into high blood pressure. You have heard of people having conditions of high blood pressure. And also we are being told to stop or reduce the amount of the cholesterol that we take in. So in this case, it is good for it to be reduced. The cholesterol will end up forming bile. Another way of lipid metabolism is we have the excess carbohydrates also being converted into fats and stored. And we said the fats in this case are stored in the adipose tissues. For example, lower part of the abdomen. Again, the fats are desaturated and then they are oxidized. That means they undergo oxidation, breaking down to yield energy. It also results into this, they are synthesized and degrades phospholipids and cholesterol. Yeah. Also, you have it synthesizes lipid transporting globulin. That means it is in the blood. So dear learner, we have come to the end of our lesson, and we can sit down and start to look at these very important questions. There are quite a number of questions which can come here, and one of them being to describe the functions of the liver. Try to note this question down. Describe the function of the liver. Then in this we can get another question, and the question will be with illustrations. Describe the process of protein or amino acid metabolism. Describe the process of amino acid metabolism. So dear learner, we have come to the end of our lesson for now. Our next lesson is going to be thermoregulation, or rather we can define it to as temperature regulation. Thank you for your viewing.